大学读博后的时候，这方面的黏膜的研究，包括我当时测序的数据也给到了经济链他们进行一个更多的分析，看跟 NK的免疫相关的反应有没有关系。所以呢，今天我们就有请到经济链给我们这方面的讲解。然后对于经济链教授呢，他是现在的教授、硕士生研究导师，广州市妇女儿童医疗中心子宫血管生物学研究室的 PI， 他的学术任职也是很多的，其中呢包括担任了 Biology of Reproduction、Journal of Reproduction Immunology 以及 Scientific Report 杂志的编委，以及非常多的国际学术的头衔。所以呢，今天我们也请经济教授给我们进行讲解。
谢谢。大家好。I'm going to be speaking in English. My Chinese is absolutely appalling. So, but all of my slides are in Chinese, so you're going to understand them better than I do. So I'm a basic scientist, not a clinician, and I am absolutely honoured to have been invited to this meeting and be included amongst so many fantastic female clinicians and researchers. So thank you very much. So for a long time, I've had a research interest in uterine natural killer cells and what they do. And today I'm going to talk about some of our own work as well as the work of others and their role in implantation and how that may lead to miscarriage or recurrent pregnancy loss. So uterine natural killer cells are the most abundant of the decidual leukocytes. So decidual leukocytes make up around 30% of all decidual stromal cells in early pregnancy, and 70% of those are the UNK cells. Traditionally, they've been characterized as distinct from the peripheral blood NK cells in that they are CD56 bright and CD16 negative. However, a couple of years ago, an extensive single cell sequencing study of the first trimester decidua started to highlight that there were 3 different populations of UNK cells in early pregnancy. We don't yet know whether or not those same populations are present in the non-pregnant and whether or not the ratios of these different populations alter in pathological pregnancies, but I am sure that those studies are currently being done. So this is just the data from that Bento Tormo paper in 2018 showing the 3 different populations. So the DNK1, DNK2, and DNK3. And these can be, so studies now need to be performed on what each of these different populations do. Up to date, we have used UNK cells as a homogeneous population, but really there now appears to be 3 very distinct populations. So what do we know about them in the non-pregnant endometrium? We know that the numbers increase exponentially across the menstrual cycle until they're at their maximum in the late secretory phase. This is data from a student of mine who was looking at menorrhagia or heavy menstrual bleeding. But if you just look at the blue bars, this is the increase, the exponential increase in the normal, as also shown here. Interestingly, in different pathologies of the endometrium, we can see differences in numbers of NK cells, of UNK cells. So in the case of our heavy menstrual bleeding, they were actually decreased, particularly in the late secretory phase. And a multitude of studies have shown that in recurrent pregnancy loss, and in recurrent implantation failure, a subset of women have very, very high numbers at LH+7. We still don't really know what the drivers of this exponential increase is, but it's considered that it's driven by progesterone. However, NK cells do not express a progesterone receptor, and it's likely an indirect effect, probably by IL-15. Further evidence of this has been shown very recently, where it's been shown that NK cells, there's a continual differentiation and trafficking in of peripheral blood NK cells, and then their differentiation to the tissue resident NK cells under the control of IL-15. But there is also some evidence of precursors within the endometrium. So it is likely that that the origin of the NK cells is both from a trafficked source from the peripheral blood and also a tissue resident precursor. The relative contributions of those different populations isn't known, and whether they give rise to different populations of NK cells also isn't yet known. A lot of the problem with this is the ethical demands of looking at first trimester tissue or obtaining precisely timed endometrial tissue is difficult, and the human is so much different in terms of its NK cell regulation than animal models, that it's harder to get a real picture of what's happening. So what do we know about what they might be doing? So we know that they are associated with blood vessel development, and in particular how muscular the blood vessels are. So this is an example of a woman with recurrent miscarriage. This is an LH+7 sample, and you can see that there is a huge number of NK cells in this sample, and this is the spiral artery, and there's a lot of muscle around this artery. Okay. We also know that when we look at the pulsatility index of these vessels, that it is altered in women with higher NK cell numbers. But whether or not this is a causal link, we still don't know. Although, interestingly, we also found in our cases of heavy menstrual bleeding, where we found a decrease in NK cell numbers, we find an association with a decrease in the development of the blood vessels in those women. So there's an association there. Whether or not it is causal, we don't know, and how it may be causing recurrent pregnancy loss, we also don't know. At the time, our speculation was that advanced vasculature and too much blood flow to the early developing placenta, which develops in a hypoxic environment, may be causing oxidative stress of the placenta, therefore the fetus, and fetal demise. More recently, in fact, just this year, a role for NK cells in biosensing of poor quality embryos has also been shown. And this role is probably more likely to be a direct impactor for recurrent pregnancy loss. We know that that in a subset of women with recurrent pregnancy loss, they're almost hyperfertile, and it may be that continual implantation of poor quality embryos, which are then later detected and lost, versus a clearance of them pre-implantation by the NK cells. but we don't yet know what the defect in new NK cells in recurrent pregnancy loss women is that would then lead to them not detecting these poor quality embryos. But it's a very interesting and perhaps in terms of therapeutics, a very, very good place to look. So then when we get into pregnancy, one of my other and probably the key research focus of my laboratory is looking at remodeling of the uterine spiral arteries. So we know that these blood vessels need to be altered in early pregnancy to allow the maternal blood flow to the placenta, which then extracts the oxygen and nutrients for the developing fetus. So these blood vessels undergo major changes from being highly muscularized and very spiral to being dilated, and eventually there's complete loss of the vascular smooth muscle cells, and the wall is replaced by extravillous trophoblast cells and fibrinoid, and there is a massive dilatation of these vessels. We also know that the early stages of this process occurs in the absence of trophoblast cells, and we and others speculated that it might be due to the leukocytes. We looked at the UNK cells, and they are surrounding the vessels, even in the presence of trophoblast cells, and when there is still vascular smooth muscle cells associated with the vessel. So we know that in vessels in both the wall and the adventitia, there's a high number of UNK cells, and also previous studies had suggested that at term UNK cells were lost. However, what we found was that there's a decrease in granulization of these cells, but not actual loss of the cells themselves. And that may well fit with these different populations that have now been detected by single cell sequencing. We also know that the UNK cells are a major source of angiogenic growth factors, and so therefore have the potential to be working on the vessels. In particular, they secrete very high levels of angiopoietin-1 and angiopoietin-2, and these differ with the gestational age from which you isolate the cells from. What happened there? I apologize. So when we started to look at this in vitro, we took chorionic plate arteries from a term placenta. This was a model for looking at a vascularized vessel, and then we could culture that in the presence of various different factors and look at how the vascular smooth muscle cells change. So if they went from their organized morphology to much more rounded up and disorganized. And what we found was that when we looked at, and I think that's what is missing in this slide, I apologize, when we looked at our UNK cell conditioned media, we get an increase in disorganization of the vascular smooth muscle cells. And when we looked at a range of different angiogenic growth factors, they could also induce this type of disorganization. But it was only when we blocked the UNK cell effect of interferon gamma did we actually reverse the effect of the UNK cells back to control levels. Okay. We also know that within spiral artery remodeling, there is a phenotypic switch of vascular smooth muscle cells, so they become less contractile and more synthetic. And during that, we can look at a range of markers. So they lose expression of H-caldesmin, myosin heavy chain, and alpha smooth muscle actin, and they gain expression of osteopontin. So these are all things that that happen in vivo that we can then model in vitro. When we looked at this, we found that both our UNK cells and our EBT could induce a phenotypic switch by reducing the expression of H-Cal-Desmin and other markers in our vascular model. Again, a range of our angiogenic growth factors could do this, and when we blocked both ANG1 and ANG2, we could reverse the effect of the UNK cell condition media. My lab is now looking into the exact mechanism by which ANG2 plays this role. So it can be speculated also that in terms of miscarriages and other pathological pregnancies that are related to an alteration in vascular remodeling, that if there is a defect with the UNK cells, too many of them, too little of them, change in the ratio of D1 of the three different populations to each other, then this very early stage of this process could be altered. And we're not the only ones who have been thinking this. There's actually another group in China, Liu et al., published a couple of papers where they have looked at NK cell derived factors, and they've picked up on interferon gamma and TGF-beta1, and both of these are also able to induce the phenotypic switch in vascular smooth muscle cells, and that they've shown that this is by activation of the long non-coding RNA, MEG3, and then its various targets. So what about their roles in invasion? So we know that the trophoblast invasion is really one of the absolutely key processes for a successful pregnancy. So a number of groups have looked at this with varying results, but in general, we see that UNK cells stimulate trophoblast invasion, which to us was incredibly surprising. And it's by a number of factors, but the key ones being IL-8 and IP-10. Also, there appears to be differences in the UNK cells derived from pregnancies with either a normal pulsatility index or a high pulsatility index. So this group that was led by Judith Cartwright had been able to show that at 11 weeks of gestation, if you took a pulsatility index of the uterine artery, that a subset of women with a high pulsatility index, even at 11 weeks, would go on to develop preeclampsia. And they did a number of studies on the UNK cells and saw quite an altered function of those from women with, even at 11 weeks, with high pulsatility index versus normal. We can also see that when we take a portion of non-pregnant myometrial artery and treat it with our UNK cell conditioned media, that it primes and facilitates invasion of trophoblast cells into the wall of that vessel. So this also suggests that the UNK cells and their role in the very early stages of remodeling are key for trophoblast invasion. So any dysfunction between this relationship of the UNK cells and trophoblast cells could lead to an aberrant pregnancy and a pathological pregnancy. So this is our working model of remodeling, which I perfectly need to update. So we know that there's UNK cells and macrophages within the wall of the vessels. We then start to see dilatation of the vessel and separation of the vascular smooth muscle cells, and they start to phenotypically switch. At this point, we then get infiltration by the trophoblast cells, and you get a much greater phenotypic switch of the vascular smooth muscle cells. Data I haven't shown, they then start to migrate away from the vessel wall, undergo apoptosis, and eventually completely lost. So what about some of the other functions of the UNK cells? Obviously, they're an immune cell. So I've only talked about their remodeling role, their tissue remodeling role, but they are an immune cell and they have an immune function. So last year, a group from the US very elegantly showed that the UNK cells, so it's been long known that UNK cells are perhaps badly named because they don't really kill, or they don't kill trophoblast cells. But in this study, they showed that Listeria-infected trophoblast cells were selectively killed by UNK cells, and it was via injection of granules or their cytotoxic granules via nanotubes into those infected cells. And a question I've been asking for a long time is the timing of infection important for the success of a pregnancy? Is there a time when you can have an infection and all the UNK cells can go off and fight that infection and the rest of the pregnancy isn't affected? Or is there a time when UNK cells are really needed to be doing their tissue remodeling role, but they have to go off and fight an infection? So their tissue remodeling doesn't get done and it impacts the pregnancy later on. And I'm not smart enough yet to work out how we test that. And I don't think I'll ever be smart enough to work out how we test that. But, so that's my big question of UNK cells and timing. When we actually, so we've always thought of UNK cells as just being involved in the uterus. However, a group in Hefei has very beautifully shown that the UNK cells also have fetal promoting effects. So they secrete factors such as PTN and OGM, which then go on to support fetal development. And this is probably under the master control of a transcription factor called PDX1. So if you want examples of very beautiful science, go and have a look at these. So we also know, coming back to pathology, so we know that UNK cell numbers are increased in recurrent pregnancy loss. We've shown that they're decreased in my brain has gone because I don't know how to read that. In heavy menstrual bleeding, there's alterations in fetal growth restriction, that they change their function and the expression of receptors in those women that might go on and develop preeclampsia versus those who won't. We also know that that through different care haplotype on the UNK cells and the haplotype of HLA-C on the fetal trophoblast cells, that can also impact the pregnancy. And there's been associations of that with recurrent pregnancy loss and preeclampsia, and actually with big babies as well. So almost that the pregnancy is too good. So just to summarize, we know there's three we now know there's 3 populations, and we need to start developing the technologies to look at their functions independently of each other, and looking at how their ratios might alter in pregnancy. We know they're involved in, they're associated with vascular development in the non-pregnant endometrium. Their direct involvement still isn't clear, but in terms of recurrent pregnancy loss, their role in biosensing is more important. They have roles in regulating trophoblast invasion and the spiral artery remodeling, alterations of all of those processes being associated with various complications of pregnancy. And I can't remember what that last one says, so I'm going to leave you to read that. And thank you very much.
谢谢 Jenny 老师教授给我们讲了一个非常生动有趣的故事，就是身体里边仅仅是一个细胞的种群，就显得这么的智慧。它从那个 NK cell 最开始它是来源于哪里，然后以及它的一些我们在基部就是女性生育和正常女性的月经周期当中不同的分布和变化，然后讲到了跟 NK cell 跟那个微血管的关系，微血管的调控。然后呢，进一步就讲到了，那么 NK cell 是一个很聪明的细胞，识别这个胚胎究竟好还是不好。然后，如果是他觉得是好的胚胎的话，那他就会通过它调节微循环的一个重塑的机制，来帮助这个胚胎进行对后面妊娠、免疫系列的调控，包括我们现在发现的一些暂时还不能解释的 NK cell 究竟是有其他什么更伟大的功能的一些现象，对我们讲。所以非常的全面，看大家有没有什么问题要Thank you for your very clear and very honest explanation for us on the NK cell. But I want, my question is, especially all the NK cell was very important for the patient devices remodeling. I'm wondering when it is occurred. It is after 12 weeks or before?
Before. So the role of the UNK cells seems to be, the role of the UNK cells seems to be prior to the role of the trophoblast. So it's very, very early in gestation. We actually found gestational age differences. So in terms of altering, just getting the vascular smooth muscle cells to separate from each other, we found that when we isolated the UNK from 8 to 10 weeks of gestation, 8 to 10, so prior to 10 weeks, then they were involved in this process. So the vascular smooth muscle cells, if we think of that they're long, interdigitated cells, and what we can see is they start to separate from each other and round up, and we could induce that before 10 weeks of, if the UNK cells were isolated from before 10 weeks of gestation.
Before 10 weeks.
Before
right?
10 weeks.
So what is the average time did you observe?
Our grouping was 8 to 10 weeks. So this was work that we did in the UK. Terminations of pregnancy are carried out much earlier here in China than they are in the UK. So we routinely looked at samples taken from either 8 to 10 weeks or 12 to 14 weeks. And I know that most of our samples now are about 7 weeks. Yeah, so work we're doing now is about 7 weeks. And we see the same effects with 7 weeks, we see the same effects. Yes. Yeah.
王教授，其实刚才王教授跟那个NK教授讨论的这个问题非常有趣。那我们请王教授大概讲一下您的问题。就是我的问题，就是因为这个NK教授他是专门做 NK 细胞在子宫内膜的这个在妊娠、病理妊娠或者是妊娠期的这种变化的。那他最主要做的就是在子宫内膜的重铸，到子宫内膜重铸跟很多产科的疾病，到正癌疟疾啊都是有关的。那刚才我问他的就是说，那血管重铸的话，它如果出现问题的话，因为他发现他重铸应该最早是什么时候开始？因为按照以前我们觉得他可能是12周以后才开始，对吧？那现在我问他说您是，他说他能看到的拿到的标本就是7周是最早的。他在7周的时候已经发现这个血管的这个下的这个血管下不是有那些平滑肌细胞吗？那些平滑肌细胞就已经开始松弛了。理论上来说，它其实松弛的话，可能就已经开始血管重铸了。因为在血管周围的重铸的时候 ，NK 细胞是聚集在这个血管周围的。实际上呢，我们是在我们看这个，我们是做的比较要前一点的，我们主要是在种植期的NK 我们做了。其实种植期的 NK 呢，我们也已经发现了，它就在血管周围开始聚集。所以应该来说，实际上我个人认为，可能它也许跟种植啊、跟血管重铸都是连在一起。所以，对，所以为什么其实跟我们产科，我们不是说妊高增的预防吃那个阿司匹林是什么时候开始？对，就是怀孕马上就吃上了嘛。Professor Lash, we discussion another important points when we use the aspirin for prevention hyperpressure disease during the pregnancy, right? So when was the perfect time to give those patients aspirin?
I don't know.
Because you know when if the vessel is modeling or as early as 7 weeks, so we should better given those medicine to prevent the hypertension of the pregnancy as early as
I think as early as possible. Yeah.
就是说它是越早越好。不一样的，不一样的。跟什么相关？跟谁？对对，他现在没有，他没有研究，他只是一定要用子宫的它是基础研究，它没有做临床研究，它只是给我们临床工作的时候提供一些信息，因为它走得很前了。我觉得这个 UNPCL 本来其实大家已经觉得在临床上可能没有太多用处了，在检测呀，或者是预测是否流产上。但是今天NK教授给我们讲的这么一个故事之后，其实又引发了大家在临床上的很多思考。其实我自己也有一个 I have another question because in my team in education before we have done quite a lot of UNPCL also, and previously the evidence said that the UNPCL is very high in both the RM, the recurrent miscarriage patient, and also the recurrent implantation failure patients.
Yeah.
But later our evidence by our research found that maybe they are slightly different, because for the RF, sometimes we hypothesize that the embryo might be okay, but the endometrium is not receptive to the embryo, so every time it is rejected. But for the miscarriage, the embryo, maybe the embryo is not okay, but the endometrium is quite receptive. So
yeah, I think like so. I worked very closely with a pathologist, a gynecological pathologist, who Judith Bulmer, who was one of the absolute pioneers of UNK cell research, and sadly she passed away last year. And when we looked at when she looked at the endometrial biopsies, and even though we knew they were precisely timed at, LH plus 7, particularly for the recurrent pregnancy loss samples, she always said that they looked advanced. So what we had wondered, and that the RIF samples looked even more advanced. So it's almost that the window of implantation had shifted, even though they were precisely timed samples with an LH surge. It was almost like, and that fits with this increased vascular development, and we wondered if they were all just markers of an advanced endometrium. So what was happening in the recurrent pregnancy loss, that they weren't quite as advanced, and so the embryo was implanting on the edge of the implantation window. But in recurrent implantation failure, where they were even further down the road, they'd completely missed that window of implantation. That's pure speculation, just from looking at the samples. And it would be interesting to use some of the window of implantation kits to look at whether or not that's actually true. But I think that they're on a spectrum, and they've shifted forwards. But in RIF, they actually missed the window of implantation, because the window of, so if you think, sorry, I do everything with my hands. So if you think the window of
就是反复种植失败和那个反复流产是2种稍微有不同的疾病模式。然后我提到这个问题之后呢，问JennieLash教授，就是其实他有没有观察到，其实NK在这里2种不同的疾病模式里面反应是不一样的。然后他就给我们分享了一个现在还非常新的观点，我还之前在学术会当中没听到，所以王平教授也觉得很有趣，就带进来讨论。是为什么呢？就是我们可能说那个在反复种植失败的患者当中，那个 NK cell 它的数量和那个胚胎组织和那个窗口期可能是有一个错位的。因为我们刚刚看到JennieLash教授在前几章幻灯片里给我们讲了那个 NK cell 它在正常的女性里是一个到了那个子宫内膜种子窗期有一个很高的陡峰升上去，然后再降下来。那么他说有可能这个种子窗期在反复种植失败的患者里头呢，它是往后移了，所以呢，它在胚胎来的时候，那个 NK 细胞并没有如期的增高，好，导致了可能胚胎种子失败。那么，然后对于反复流产的患者呢，可能是这个峰提前了一点来了过后呢，它有一个峰，但是这个峰可能比较小，也不足够支撑胚胎继续生长下去，因为那个 NK 的重塑的作用就没有体现出来。所以呢，导致了后期的流产。这个观点是非常新的，当然呢，也肯定期待更多的数据出来，所以我觉得非常有趣。谢谢大家。